What is mitosis? Is how non-sex body cells reproduce. It is asexual, meaning that there is only one parent. The cell divides into two identical diploid cells with two full sets of 46 chromosomes each. The function of mitosis is to grow, repair, or replace tissue. What is meiosis? Meiosis is how sex cells reproduce. Meiosis is sexual, meaning that there is more than one parent. This produces 4 haploid cells, with one full set of 23 chromosomes each. The function of meiosis is to create genetic diversity through sexual reproduction.
What is the cell cycle? The cell cycle is a series of events that occurs as a cell grows and divides. The longest phase of the cell cycle is interphase. During interphase, the cell grows and goes through its normal life functions. The shortest phase is mitosis, which is when the cell divides. Throughout the G1 period, the cell lives its normal life. Then something happens, like a cell needing to be replaced, that signals the cell that it is time to prepare for mitosis. During the synthesis, or S period, the cell duplicates its DNA. It doubles its 46 chromosomes and ends up with 92. In the G2 period, the DNA is checked for errors. An error such as DNA mutation could cause cancer. Finally, the mitosis period begins.
This is what a cell looks like during interphase. This is the nuclear envelope. Inside it is chromatin. These are centrioles. At the beginning of mitosis, they start moving to the edges of the cell. In prophase, the centrioles begin to create spindle fibers. The nuclear envelope begins to dissolve, and the chromatin turns into chromosomes. This is a chromosome. Chromosomes contain genetic information. In the center is a centromere, and a chromosome is composed of 2 sister chromatids. Next comes metaphase. The chromosomes line up in the middle of the cell, and the spindles attach to the centromeres. In anaphase, the chromatids are pulled apart. During telophase, the cell cleaves, and nuclear envelopes begin to develop. 2 sets of 46 chromosomes, and the spindle fibers disappear. After mitosis, the cell is fully divided into 2 daughter cells. These cells are clones and genetically identical. They are diploid cells because each cell has 2 sets of chromosomes, a total of 46 chromosomes per cell.
Meiosis is different. After the cell replicates its DNA during the interphase, the cell begins meiosis 1, starting with prophase 1. In prophase 1, the nuclear envelope dissolves. Centrioles move to opposite sides of the cell, and spindle fibers are formed. This is into chromosomes. There are 2 of every chromosome, one from mom and one from dad. Homologous chromosomes pair up. Then crossing over occurs, which is when pieces of non-sister chromatids trade places. In metaphase 1, spindle fibers attach their homologous pairs, which line up along the middle of the cell. In anaphase 1, spindle fibers pull apart the homologous pairs. 1 chromosome of each pair goes to one side of the cell, while the other goes to the opposite side. In telophase 1, the cell cleaves. The spindle dissolves and nuclear envelopes form. In cytokinesis, we end up with 2 haploid daughter cells with 46 chromosomes each. Each cell has a random mix of chromosomes, 1 from each pair. These cells then move on to meiosis 2 without duplicating DNA.
In prophase 2, the nuclear envelope dissolves. The centrioles move to opposite sides of the cell, and the spindle begins to form. In metaphase 2, the spindle fiber attaches to chromosomes, which along the middle of the cell. In anaphase 2, spindle fiber pull apart the sister chromatids, which then move to the opposite sides. In telophase 2, the nuclear envelope forms and the chromosomes uncoil into chromatin as the cell cleaves. In cytokinesis 2, we end up with 4 haploid cells with 23 chromosomes each.
Differentiation is the process through which a non-specialized cell, such as a stem cell, develops a specialized form and function. Differentiation takes place after asexual reproduction and during embryonic development. It is important because it also occurs during the renewal of tissues, which means that differentiation keeps making new cells when you lose cells. What happens if something goes wrong? If a problem, such as DNA mutation, is missed in the G2 phase, which is when the DNA is checked and repaired, this can cause conditions like Down syndrome, sickle cell anemia, and cystic fibrosis. If the DNA is not repaired, then that mutated cell will continue to reproduce, creating more and more mutated cells. When the cell cycle is no longer regulated, cancer occurs. Cancer often results in dangerous and deadly tumors. Humans have 23 chromosomes in one set, but this isn't the case for all animals. What makes an organism unique is that they have different types of genetic traits. If all organisms had a different number of chromosomes, then some would have millions of chromosomes, and a cell could not contain all of them. For example, chickens and dogs both have 76 chromosomes. Why aren't I identical to myself? Each of these cells, which will be either a sperm or an egg, contain different genetic information despite coming from the same parent. You don't look exactly like your siblings because the DNA is free at random. The chances of having the same exact baby are 1 in a trillion, because with 23 chromosomes, there are a trillion possibilities.